we're going to continue on where we left off last time. So last time we were talking about myasthenia gravis, and myasthenia gravis is an autoimmune condition. And with an autoimmune condition like that, you're going to see destruction of the nicotinic receptors at the motor end plate. And so recall that with myasthenic type symptoms, you're basically going to get weakness of the muscles. Now, if you remember with Lambert-Eaton myasthenia, that was a little bit different because with Lambert-Eaton, you basically regain function as you're using your muscles. So it gets progressively better as you're exercising or using your muscles versus myasthenia gravis, which is the opposite. You wake up in the morning, you're feeling fine, but then as you progressively use your muscles more and more, you're going to see more and more weakness. How do you test for it? You can again do that tinselin test, and edrophonium basically is an acetylcholinesterase inhibitor. And so if you're destroying those nicotinic receptors, then you're going to see that if you have acetylcholine, hitting those receptors, it's not going to hit enough of those receptors to actually result in an action potential in the muscle. Now, if you have an acetylcholinesterase inhibitor, acetylcholine can linger longer within the synaptic cleft. And so that's one of the ways that you can treat it. Now, I made a change on your guys' lecture slides. The pyridostigmine, which is one of the types of drugs you could use, it said on there was an anticholinergic. It is not an anticholinergic. So you can scratch that off your slides. It is actually an acetylcholinesterase inhibitor. If it was an anticholinergic drug, it would basically block the ability for acetylcholine to work. So I apologize for that little mistake on your lecture slides. So in other words, you basically prevent the degradation of the neurotransmitter. If you prevent it from degrading, then it's going to be able to linger in the synaptic cleft for a longer time. And that's how it helps to basically treat the symptoms of myasthenia gravis. Now, the thing about myasthenia gravis is that it's not curable, right? So you can only really treat it symptomatically. And that's basically it for myasthenia gravis. So now let's switch gears and talk about other compounds that can affect the acetylcholine transmission, whether it's acetylcholine release or the calcium that's going to be involved in that. So the main one that we're going to be looking at is going to be botulinum as well as tetanus. Now, these are going to be derived from Clostridium organisms. So Clostridium is a genus of bacteria, and there's a bunch that are clinically relevant, but the ones that we're going to focus on are Clostridium botulinum as well as Clostridium tetani. The other compounds that your textbook talks about that I'm not going to hold you guys responsible for are latrotoxin. Do you guys know where latrotoxin comes from? That's fine. Latrotoxin actually comes from black widows. So that's actually the black widow toxin. And so latrotoxin is going to do something kind of similar to what botulinum does. Basically, it's going to affect acetylcholine release. And what you get with this is you get too much acetylcholine release with latrotoxin versus botulinum and tetanus. It's basically going to be a little bit different mechanism of action. So Clostridium bacteria causes botulism and tetanus. What they do at the cellular level is they affect the snare proteins. And the snare proteins are involved in the fusion of the vesicles to the presynaptic terminal. And so basically it's going to affect the ability for acetylcholine to get released into within the synapse. The biggest concern with these types of toxins is that you basically get the inability to control breathing, right? So with botulism, you get paralysis of the muscles. So you get flaccid paralysis, which of course, if you paralyze the diaphragm, you're going to stop breathing. The opposite is true with tetanus. You're going to get spastic paralysis with tetanus. And so with spastic paralysis can also affect the way that you breathe. So if it paralyzes your diaphragm, you're not going to be able to breathe properly. So that's going to be one of the major ways that these toxins kill you if left untreated. Yes.
What do the snare proteins do again?
Snare proteins are going to allow for the vesicles to attach. So remember, within the vesicles is where you have neurotransmitters. And neurotransmitters are going to be hanging out in the vesicle until you get stimulation of the neuron so that you can actually release the neurotransmitters from those vesicles. And so one of the mechanisms for the vesicles to attach themselves to the presynaptic terminal is with the use of these different types of proteins, snaps, snares. So if we go back to what we were talking about just the other day, so here, for example, this is a vesicle. This giant green blob is a vesicle, and all of these little diagrams of like structures, those are all different types of proteins that are involved in vesicular transmission. So it's allowing for vesicles to move in different areas. It's also allowing for the vesicles to attach themselves to the presynaptic terminal to allow for the vesicles to then release their neurotransmitters into the synapse. So there's tons of different proteins. Again, snaps, snares, those are going to be some of the major ones, but there's other ones as well. Synapsin, NSFs. I'm not going to hold you guys accountable for all of these different types of proteins, but there's a bunch of them, and they all do different types of functions. But at the end of the day, the whole purpose of these is to allow for the vesicles to release neurotransmitters through exocytosis. So they bind to the, like it shows over here in this image, you got the vesicle binding to the presynaptic terminal, the phospholipid bilayer, and when they bind, they're able to open up and allow for the contents to be released through exocytosis. So you see the binding using different types of proteins like synaptobrevin, synaptogammin, and then eventually you get fusion and then release of neurotransmitters. And so botulism as well as tetanus can potentially affect that whole process. So let's go back to botulism and tetanus. So here's botulism and tetanus affecting the snare proteins, and basically they're going to cleave those different proteins. There's going to be also like proteases involved in this whole process. Long story short, though, it interferes with the ability for neurotransmitters to get released. If you can't release neurotransmitters, you're not going to get proper functioning at the muscles. Again, can cause weakness and death. So let's now talk a little bit about tetanus and the signs and symptoms of tetanus. How do you get tetanus? Rust. Okay, so is that correct? Is that how you get tetanus?
Isn't it like dirt?
Yeah, so it's going to be something found within the dirt. So these are spore-forming bacteria, Clostridium. And so if you have a spore-forming bacteria, you're going to find those in all sorts of environments. Now, with tetanus in particular, it's an anaerobic organism. It doesn't like oxygen. So when it's hanging out in the soil, it's going to be exposed to oxygen. What does it do to remain dormant and protect itself from the environment? It's going to form spores. So the spore is basically the way it can be dormant for a very long time. And spores can exist in the environment for like up to 100 years. So they can stay in the environment for a very long time. A spore is basically just a bunch of like calcium, a bunch of glycocalyx, which are different types of proteins, and that basically protect the DNA. So the spore forms a protective coat, protects the DNA. Once the spore gets introduced into a hospitable environment, for tetanus in particular, an anaerobic environment, then it goes into a vegetative state. And so it goes from spore to a vegetative bacteria. And that's what releases the toxins, that's what now can reproduce, and that's when it's going to go about its normal metabolic activity. And so why is it associated with rust? Because when iron is exposed to oxidizing agents, especially in like soil, you're going to see rust formation. So it's not the rust itself, it's the fact that it's like dirt, soil, oxidizing environments, right? That's why you're associating it with rust, but also nails, because deep penetrating puncture wounds introduce the bacteria into an anaerobic place, right? If you just get a superficial scratch, you're not going to get tetanus, right? Because it's exposed to oxygen. However, if you have a deep penetrating wound with like a nail, now it's going to go into deep layers of the tissue, and you're going to see more anaerobic environment conditions in a deep flesh wound. And so that's why we're concerned about deep tissue injuries, right? That's why if you get into an accident, you'll probably get some sort of like either tetanus immunoglobulin, which can help protect it in the short term, or you'll get the TD vaccine, which is the actual it's the vaccine that allows for active immunity. So this happened, like, for example, I got into an accident about, almost 5 years ago now. It was like my best buddy's birthday. We were celebrating. I was ready to go home and take an Uber, but he was like insisting that we took scooters. And so and I'm like, I don't know if this is a good idea. He's like, come on, man. There was 3 scooters. There was 3 of us, and there was 3 scooters. And he's like, dude, it's perfect. There's 3 scooters right there. We're on Mill Avenue. And I'm like, God, this is such a bad idea. So then, like, in my, I wasn't thinking straight. Let's put it that way. And I got on a scooter. Next thing I know, I'm like in an ambulance. I was like, what the hell? I thought I was dying because like I just had this, like, I fell off my scooter. And I turned into like an eggplant. Like my face was like, it was awful. I thought I was like never going to look okay again. Huh? No, I just fell. I think I was trying to turn a corner and I just plopped. And so I woke up in an ambulance, and then like I was reading through my medical reports when I got discharged. I got like sutures and stitches and stuff. And I saw in my reports, they said they administered a tetanus vaccine because I had like injuries, like pretty nasty injuries to my face. So they gave me a tetanus vaccine, and that will happen. Like if you're unconscious, especially, and you can't tell the paramedics or the doctors, or whatever, that you've had a tetanus vaccine within the past 10 years, they'll just give it to you if you're unconscious because they don't want to take the risk. I don't even remember. Was it bad? I don't even remember that. Yeah, some vaccines can be very painful. Like, don't get syphilis because the penicillin injections are extremely painful. I had a patient that had secondary syphilis, and so if you have secondary syphilis, you get 3 intramuscular injections in the gluteals, and 1 every week. And you can barely sit down. It's extremely painful. So yeah, penicillin shots are remarkably expensive, or not remarkably painful, but I'm allergic to penicillin too. So yeah. Anyways, so it's an anaerobic bacteria. If it gets introduced into your deep tissues, that's when it's concerning. And then it can actually start causing some issues. This painting over here, this is a depiction of opisthotonus. And opisthotonus is where you have rigidity and hyper flexion. So you have flexion of all of your muscles. So it's like a spastic paralysis. This painting was done by Sir Charles Bell, and it's showing basically a soldier that got exposed to tetanus. A lot of times back in the day, in the battlefield, soldiers would get tetanus, and that was usually what would cause death in some of those individuals. And this can be so intense that it can actually fracture your vertebral bones. So you can actually get vertebral fractures. You can even get large bone fractures too. So this is no joke. If left untreated, it will cause some serious issues. That's opisthotonus. And this is opisthotonus that's affecting a couple of young kids. This is a baby. This is a neonate with opisthotonus. How do you think this kid got opisthotonus? Do you think he stepped on a rusty nail?
No. His mom had us.
That's a really good guess. It's not from the mom.
Used in the hospital?
Yes, close. What happens when a child is born?
Oh,
Cutting the umbilical cord. So this was transmitted to a neonate when they were cutting the umbilical cord using contaminated equipment, right? Contaminated scissors, contaminated scalpel blades. So yeah, this is really, really nasty stuff. So not good.
And there's no really cure.
Well, yeah, you can try to give the patient muscle relaxants. You can try to give them sedatives so it can reduce anxiety and things like that. But it will eventually run its course, and it will eventually, regain function. But it takes time. But the toxin, once it disseminates systematically, it's going to cause lockjaw. And how does it work? It basically prevents glycine and GABA to be released. And we're going to talk about our neurotransmitters in the next section of these presentations. We're going to talk about your inhibitory neurotransmitters. GABA and glycine are both your principal inhibitory neurotransmitters, particularly GABA. So if you can't release GABA, what happens to your muscles? They're going to be overly stimulated, right? They're going to have too much acetylcholine taking over, acetylcholine binding to nicotinic receptors is going to cause spastic paralysis. So you get these uncontrollable muscle spasms. Other things that can cause death, not just paralysis of the diaphragm, but also these individuals tend to aspirate. So they get contents from the GI tract into their respiratory tract, and then they get aspiration pneumonia, and that's usually what kills the patient. So again, Clostridium tetani, that's going to be the main cause of this. The toxin is called tetanospasmin, and tetanospasmin is an AB type toxin. AB, it's so annoying because B is the thing that attaches to the cell. A is what gets brought into the cell. It should be the other way around, but usually these types of things are named in order of discovery. So the A component was probably discovered first, and then the B component. But anyways, not important for this class. Just know that these toxins attach to the cell, enter into the cell, and that's when they start causing some serious issues. Now, you guys have all probably had the DTaP vaccine. DTaP, that's going to be diphtheria, tetanus, and pertussis. So that's a very, commonly used vaccine. That's part of like the normal vaccine schedules for kids. As you get older, you might get the Tdap instead, so it's age dependent. And if you're much older, you might just get the TD vaccine. The TD specifically contains the components to target the tetanospasmin toxin. So different people might get it at different types of vaccines at different age ranges. Yes?
I might be dumb. Did we talk about botulism?
We're going to talk about botulism right now, actually. Yeah, not yet. So here's Clostridium botulinum. Clostridium botulinum, also a gram-positive organism. Don't worry about, by the way, for this class, don't worry about gram-positive, gram-negative. That's more like microbiology. You'll be tested in that kind of stuff in microbiology if you haven't already taken micro. You took it already?
2 years.
Oh, okay, you took it a while ago. So this is like a refresher for you. This does the opposite, though. This causes flaccid paralysis, and it's going to affect acetylcholine release. So tetanus affects GABA and glycine, whereas botulism affects acetylcholine in particular. And so if you can't release acetylcholine, you're not going to be able to stimulate those nicotinic receptors at the motor end plate, and that's going to cause flaccid paralysis. Now, here's an adult with flaccid paralysis from botulism. And then here's a kid, another young individual. So first of all, how would an adult usually get botulism toxicity? And it happens occasionally. It's like a handful of cases every year here in the United States. Has anybody done any like home canning or home jarring, like jarring your own like fruits and meats and stuff? So if you improperly can at home, then that can increase the risk of getting botulism. So again, botulism is spore-forming bacteria, and those spores are very, very robust. They can protect the bacteria and the DNA from harsh environments. So botulism is going to be spore formation, and to be able to kill the spores, it's very, very difficult. You have to autoclave, basically. So pasteurize when you're making like home canned goods, usually using jars. You use a giant pressure cooker and you crank it up to 15 psi, and you have to do that for about 2 hours or so. And then that usually can eliminate most of those spores. But if you don't do it to a certain temperature or a certain pressure or for a certain period of time, some of those spores might still survive. And usually that's how you get botulism, is from poorly canned goods. You can also get this too if you like go to the store and you see a can that looks, maybe it's like bulging or maybe it's like dented. Stay away from those. Right? Those cans are probably contaminated with something. Could be botulism. How do kids get it?
Wait, is it from the can or the?
It's from the stuff in the can. So the food was contaminated with botulism spores, and then you didn't properly pasteurize the food or the contents.
That's why they said to wash the vegetables and fruits.
Well, that's not so much. That's not so much the case. It's more so.
They say that it's like.
If it's bulged, yeah, you avoid it altogether. But it's mostly canning, right? Not necessarily like not cleaning off your foods too well. However, this one is probably associated with food contamination. What kind of food do they tell moms not to feed their children? Honey. Why? It's because of this. Because honey is often contaminated with botulism. Now, a kid, when they eat honey, their immune system is not that robust, right? For us, if we eat contaminated honey, our immune system can probably kill off the bacteria that's in there. But for a young individual, like a very young, like infant or a neonate, their immune systems are not that good. So you want to avoid giving honey to young children because of that. So what's the mechanism that's going to be involved in the ability for neurotransmitters to be released? These are a couple of steps that are involved in this. So you have a thing called clathrin. So they got these like 3 little hooks that allow binding to the presynaptic terminal. Then you have the formation of a vesicle with the use of dynamin. And then you also have actin filaments, which help to basically guide and move those vesicles within the neuron. And then you have the release of dynamin, and then you have your final product, which is the vesicle. This is going to be energy dependent, so you have to have ATP for this whole process to take place. But clathrin, dynamin, those are going to be some of the most important proteins in the formation of vesicles. So here's another image depicting that. So you got your clathrin structure, which has those 3 different hooks, which allows for the formation of a vesicle. And once the vesicle is formed, that's when you can actually start getting things like neurotransmitters within that vesicle. Now, what kind of transmission are we talking about when we discuss neurotransmitters? So there's a couple types. So there's going to be ligand-gated ion channels. So think about the neurotransmitter as acting like a ligand. It binds to the protein to allow for ions now to move through, right? So you get a conformational change within the channel once it binds to the neurotransmitter, and then you can get ions coming through. And this is a pretty fast process. It happens in 1 to 2 milliseconds as soon as you get attachment. Then you have metabotropic receptors. And metabotropic receptors are going to be a little bit slower because they have to use a G protein coupled receptor or a G coupled protein process. So you have like a downstream cascade that happens within the cell once you have once you have neurotransmitter binding to the receptors. Again, this takes a little bit longer than ligand-gated channels. And then you also have a bunch of phosphorylation events that happen within the cell. So it's like, think about your second messenger systems. Right? This is kind of similar to that, right? Similar, yeah, similar. You have like a bunch of downstream cascades that affect the actual, like you get the actual result of the neurotransmitter binding. So here's a ligand-gated ion, very simple, basic type of mechanism. You just bind to the protein like a lock and key, then it opens up the ion channel, and then you can get ions moving freely into the cell. Versus G-coupled protein, metabotropic type receptors, you have a whole entire downstream cascade. Eventually you get the same type of thing. You get ions moving downstream into the cell as well, but you have to have a bunch of these intermediary steps. So that's what we're talking about when we discuss G-coupled proteins. Acetylcholine is going to be involved in those ligand-gated type channels. So whenever you have binding at the motor end plate, right? So end plate currents are going to be stimulated. And so once you have, example, in the neuromuscular junction, acetylcholine binds to the nicotinic receptor, then you can have ions coming into the cell to allow the muscle cell to depolarize. This also happens when you have a synapse between 2 neurons, similar type of thing. So you get influx of sodium, and then you also get an efflux of potassium. And that's how you get the end plate current, or an EPC. And so here's a couple of different experiments that they did using a patch clamp technique. Remember, with the patch clamp technique, a little bit different than the voltage clamp technique. Patch clamp, you're literally just taking one single ion channel, right, and then seeing what kind of conditions can affect the the functioning of the ion channel. And so with this one, they were looking at the postsynaptic muscle cell. So this is specifically the ion channel on a muscle cell, and they were seeing what kind of things affect acetylcholine, what kind of things affect the ability for the channels to open up. So this is mostly FYI. I just wanted to include that in there from your textbook because it's different experiments that show how these acetylcholine receptors actually work and how they affect ion channels. Yes.
So just really quickly, so acetylcholine binds to the receptor, and then the ligand gated channel opens.
Yes.
And then more acetylcholine.
No. Ions move into the cell.
Okay.
So acetylcholine binds to the receptor, it opens up the ion channel, then ions come into the cell, and then it allows the cell to depolarize.
Okay.
Right? And then for the muscle, the depolarization, you have sodium coming in, allowing for depolarization. That depolarization signal gets sent all the way down to the sarcoplasmic reticulum, allowing for calcium then to be released, right? You have like the ryanodine receptor, which is voltage gated, that allows for calcium to be released from the sarcoplasmic reticulum, and then you can get contraction of the muscle. Right, so this is like, it's all involving like a cascade. Right? It starts with the neurotransmitter binding to the nicotinic receptor. Sodium comes in, depolarizes the muscle cell. That allows for calcium to be released. So huge sequence of events. So cool stuff. Lots goes into this. And again, this is not a math class, so I'm not going to make you do any calculations, but this is an example of how they would calculate the end plate current. So EPC is the end plate current. You have conductance, which is G of whatever substance. For this case, it's acetylcholine. Then you have the membrane potential, which is VM, and then you have the reverse potential. And the reverse potential is basically what allows different ions to move in the opposite direction, which allows for depolarization. And so voltage, so the reverse potential is defined as voltage at which direction of the current changes from outward to inward. And that's basically what allows for the actual depolarization event. And so with acetylcholine, acetylcholine channels are going to be permeable to both sodium as well as potassium. And there's been experiments done where they, if they put the voltage at 0 millivolts, you're going to see that basically the neurons stay at a standstill, right? You don't have any movement of current because at 0 millivolts is when you have equal permeability to both sodium as well as potassium. So here's a couple other experiments where they were stimulating neurons and then seeing what kind of effect it had within the actual muscle cell. And so at 0 millivolts, you're getting 0 movement, right? So you basically achieve equilibrium between potassium and sodium ions. And then there's different voltages that are going to be associated with the movement of potassium. They're associated with sodium in particular, as well as chloride ions. And I'm not going to test you on the different voltages that are specific to each of these ions. But if you're curious, these are some of the values. So if you get close to about negative 100 or so, you're going to get movement of potassium. When you go above 50, around like 60 or so, that's when you're going to start seeing movement of sodium. So, in other words, you're trying to change the directions of the ions moving. So if you lower the concentration of sodium, so that's what they're talking about over here with the brackets, right? So if you lower the overall concentration of sodium, it's going to make the it's going to make the resting potential a little bit more negative, and so it's going to make the movement of sodium go in the opposite direction. If you increase external potassium, that's going to make the resting potential or membrane potential of potassium more positive, and it's going to basically allow for the opposite direction of movement for potassium. Long story short, though, with this, I'm not necessarily going to like assess you guys on like reverse potentials, but just know that with a acetylcholine channel, when it gets activated, it's going to be permeable to not just one type of ion. It's going to be both sodium as well as potassium. And so here's an example of this as well. So you have reduction of external sodium. It's going to make the sodium reverse potential shift, right? It's going to make it move in the opposite direction. And then if you manipulate the concentrations of potassium, it's also going to make potassium movement in the opposite direction. So basically, that's kind of what reverse potentials mean. So you're like basically stimulating the axon or stimulating the cell so that you have opposite direction of current. And again, at 0 millivolts, that's when you have equal currents. At 0 millivolts, the current's going to stabilize. Yeah, so it's basically going to stabilize the currents at 0 millivolts. And that all has to do with the acetylcholine ion-gated channels. So, the action of neurotransmitters. It's to drive the postsynaptic membrane potential towards the reverse potential of an ion. That's the ion channel being activated. So when you activate the ion channel, you're going to want to see that ion moving in an opposite direction. So during depolarization, so here's another way to look at it, the postsynaptic membrane potential is driven towards the reverse potential for sodium. That's during depolarization. So remember, you depolarize the cell, you make the cell become more positively charged, right? And then that's going to basically stimulate sodium in particular to move. When you repolarize the cell and you bring it back towards a negative charge, that's going to basically force potassium to move, right? So it's going to affect the driving, consider these like driving forces, right? The charges are going to be the actual driving force for the movement of the ion. So more depolarized, more positively charged, you get sodium movement. More repolarized, more negatively charged, you get potassium movement. Do you have a question?
Yeah, so the movement of the ions, are we specifically talking about like sodium coming into the cell so it's more prominent outside?
Yeah, basically.
And then vice versa.
Yeah, so that's the reverse potential where you're going to have sodium coming into the cell when it's depolarized, potassium leaving the cell when it's being repolarized.
So you're basically just adding a name to what we know.
Basically.
Basically.
To simplify it, yes. Depolarization, sodium coming in. Repolarization, potassium leaving the cell. So these are just different experiments basically describing the same type of phenomenon. Good call on that. Love it.
Sometimes I have like aha moments.
Yeah. Again, at 0 millivolts, equal movement, right? So sodium and potassium are going to be moving in equal directions and equal quantities. So it basically stabilizes. All right. Now let's talk about excitatory versus inhibitory action potentials. So for glutamate is going to be one of the most important excitatory neurotransmitters. And when you have stimulation of glutamate, that's going to allow for sodium to come into the cell. And when sodium comes into the cell, it stimulates the cell, right? It allows for activation and depolarization of the cell. Versus inhibitory postsynaptic receptors, GABA is one, glycine is another. These are going to affect chloride movement as well as potassium as well. So if you have chloride ions coming into the cell, that makes the cell more and more negative. So that hyperpolarizes the cell, and that prevents the cell from being able to become activated, right? It prevents depolarization. If you also push potassium out of the cell, that's also going to make the cell more negative. And so if you stimulate potassium efflux, then that's going to, again, hinder the ability for the neuron to actually fire. And so, long story short, inhibitory postsynaptic potentials are going to generally involve chloride ions coming in or potassium ions coming out of the cell. Versus excitatory postsynaptic potentials allow for sodium to come into the cell to allow for actual depolarization. So we're going to talk about those types of neurotransmitters shortly. So here are the things that affect the ability for the excitatory postsynaptic potentials as well as the inhibitory postsynaptic potentials to actually affect the neuron. So space, in other words, how far are these potentials occurring from the axon hillock, right? Because remember, with the axon hillock, that's right before you get to the actual axon itself, right? You have to think of it as like a funnel, right? You get all those signals eventually reaching the axon hillock as a funnel. Once that area gets reaches threshold, then you actually get the depolarization down the entire axon. So if you don't reach thresholds, you're not going to get any signal whatsoever. Also, time. How many of these signals are being sent to that receiving neuron, right? So if you have it spaced out over longer time, you're not going to, reach threshold, right? If it's spaced in a shorter period of time, then it's going to maximize the ability for that cell to reach threshold and then get a depolarization. And then you get a combination of all the different signals. So you have neurons that are going to be receiving inhibitory signals, excitatory signals all the time, right? And it depends on how much of the excitatory signals they receive. That's when you actually get the depolarization of the neuron. So you'd have to add it up, subtract the ones that are inhibitory, add up the ones that are excitatory, and then once you reach threshold with the combination of all those signals, then you can actually reach threshold. So here's a receiving neuron over here, right? So you got the axon hillock is going to be this area between the cell body and the axon itself. And the dendrites are going to be receiving signals from all sorts of different types of neurons. Again, inhibitory and excitatory. And so you basically add up how many excitatory signals they receive. And again, if they reach threshold, that's when you indeed get the signal. And then again, convergence, you have a bunch of these synaptic signals converging to one neuron, right? That's kind of what's going on over here. And again, in order for that receiving neuron to depolarize, it has to reach threshold. And so here's the overview of all the different steps, all the series of events that have to take place. So you get the neurotransmitters being released, whether it's excitatory versus inhibitory. They bind to the receptor. The ion channels close, right, or open up, depending on what type of receptor binding you get. Then you get conductance, right, movement of the ions. Then you get the changes in the postsynaptic cell potential. And then you either see inhibition or excitation of the cell, and then you have to do a summation. You have to add up all those excitatory signals to actually reach threshold to get a full-blown action potential. Yeah.
When you're adding the signals, it's basically the membrane potential or voltages.
Yeah, it's chloride ions coming in, it's sodium ions coming in, it's potassium ions leaving the cell, right? And depending on the overall charge of the cell, once it reaches that threshold voltage, then you can actually get a full-blown depolarization. But you add up all the inhibitory plus excitatory signals, and if you actually get an excitatory signal that's enough to reach threshold, that's when you get depolarization. Pretty basic stuff. All right, let's do a couple quizzes questions. Let's do maybe like 3 or 4.